So let's go ahead and get started. I have several things that I have written on the board. Today we're going to talk about phospholipids. This shouldn't take us very long. This is just the end of that chapter 8 stuff. But it's an important ingredient in a lot of foods. And so I wanted to address that. And then we'll just broadly discuss different types of vitamins. I'm not going to go into as much detail as I normally do on the types of vitamins, partially because remember your assignment for the day you're not here is to watch the movie Vitamania. And there should be a link on Canvas. It will require a password, and the password is written there right next to the link. So you'll watch that, and then there are some questions to answer. And rather than doing it in a discussion, you're just going to submit your answers to me. And you can handwrite them, you can type them, I don't care, but upload them to Canvas. And then we are going to do an activity where we are going to see how antioxidants actually stop oxidation using glow sticks. And we're going to do kind of a summative activity looking at ingredient labels and seeing, okay, what are all of these weird names that we see in foods? So after that, I have several things that are kind of coming up. Hopefully most of you are getting close to finished with your infographic. Some of you have remembered to upload your rough draft. And mostly I'm just giving you credit for uploading the rough draft. You're either getting 4 or 5 points for giving me the rough draft unless it's in really bad shape. But I am putting comments like it looks like you're missing the graph, or it looks like you haven't done X, Y, or Z. So just because you get 5 points on the rough draft doesn't mean, oh, it's perfect. It just means, yes, you've turned something in. And then, let's see, I haven't posted homework 6 yet. It should be on the shorter side, just a few questions. And remember, it's covering 2 chapters, chapter 7 and chapter 8. And the last thing is that study analysis that I kept putting off. And I decided, because I'm not getting into vitamins as much as I would like to, I would give you 2 options. So you can either just look up a study like you did the first time, and kind of like I explained to you before. And with that, you can, you're just going to summarize it rather than putting everything in bullet form or just answering questions. You're going to write paragraphs, work on those writing skills. The other thing you can do is I posted several either audio or video files that talk about experiments that were done in order to discover what vitamins did. And so you can use one of those as the basis of your study summary. So you can write a summary of what that was. Do remember that if you choose that option, you do still need to say where you got your information from. Provide that, and that is going to be due the day of the final. So you don't have to worry about getting that done in the next couple of days. You got a good week to do that over the weekend, so you have some time on that. What I will say about that, it is my intention, since the final is mostly going to cover these last couple of chapters, but I'm going to do a couple of summative things. The way I'm going to test the experimental design stuff we talked about at the beginning, the independent variable, dependent variable, those kinds of things, is I will give you a study based on one of the vitamins. So if you do that, then there's a chance that you'll hit the one that I'll put on the final exam. And there's several to choose from. So any questions about what's coming up? Okay, I did some fiddling around on Canvas, and I reduced the amount that the test is weighting towards your grade. And for most people, that's not affecting your grade a whole lot. You're kind of doing evenly throughout, but I think it's helping some people who were having trouble with some of the tests. This last test was a little bit more rough than the first test. So by spreading more of those points to the participation type stuff and less of the points towards the exams, I think it's helping your grades. But if you have questions about that and how it affects your grade specifically, let me know and we can go over your grade individually. Okay. There we go. Okay, So one of the first lipids that we talked about was triglycerides. And so we said with triglycerides, we have these chains of fatty acids that attach themselves to a glycerol molecule. And when they do, the hydrogen that's part of that glycerol goes away and we get these ester linkages. So this is an ester, this is an ester, this is an ester. And so this molecule on the left here has 3 fatty acids shown in blue, green, and red. And we've got this blue one is saturated. How do we know it's saturated? What are we looking for? Definitely a good thing to know for your final exam. There are no double bonds in here. So this one's saturated. And then this one is mono unsaturated, meaning there's one double bond on this chain. And this one is poly unsaturated. And both of these, the double bonds occur in this kind of trapezoid type area. And so that's an indication to us that they are cis unsaturated. Okay, so this is going to be nonpolar. And this is going to be the fats that we normally think of as lard, as oil, those kinds of things. Now, this looks very, very similar. And this is a phospholipid. So we can see it has 2 of these long chains that are very similar. And on any triglyceride, any phospholipid, these could be any combination of whatever fatty acids. They could be all saturated. They could be all unsaturated. So the exact chain is going to depend on which specific molecule you're looking at. And so that's why we talk about them as a group rather than a specific chemical. But the difference is down here, we have this whole region that where the oxygen, instead of being attached to another carbon, is attached to a phosphorus. So we call this a phospholipid because there's a phosphorus here. Now, phosphorus is over closer to oxygen. And remember, the closer we get to oxygen, the more electronegative. And electronegativity is tied to that polarity, that on organic molecules, when we have an electronegative atom, we're going to get some polarity. So we have the polarity there, plus we've got all of these oxygens and a nitrogen over here. So all of this stuff is very polar. And we do have some carbons here, but instead of long chains, they're just real short. So what that means is that if I had a bunch of water molecules all over the place here, these water molecules would keep bumping into these fatty acids and have trouble getting into this area where the oxygens are. But the water molecules over on this side would be able to mingle over here and really surround those oxygens. And so this is what we call an emulsifier. That was one of our words from early in the semester. An emulsifier has a fatty part or non-polar part, and it has a polar part, like this one here, okay? And so this one is less likely to react to form ester linkages the way that the individual fatty acids do. And so it's a pretty stable molecule. And where they're typically found is going to be in our cell membranes. And so we'll talk about that in just a minute. So a lot of times we'll talk about these as diglycerides, di meaning 2. So that's because we have the 2 fatty acids. And a lot of times, rather than drawing all of this out, we'll draw a circle with 2 lines coming down. So we talk about this circle part is where the phosphorus is and all the oxygens. So all the ester linkages are there and everything that's possibly polar is right up there together, okay? So this is our head, A is our head, and then the bottom part is our 2 tails, okay? And the 2 tails are non-polar. So if I had a couple of these phospholipids that were near each other, the heads would hang out together because they're both polar, and then the tails would hang out together because the tails are both non-polar. So they would just sort of line up with all of the heads facing in the same direction, all of the tails facing in the same direction. All right, so the head is what we call polar. Another word for polar is hydrophilic. Hydro means water. Philic means to love, like Philadelphia is the city of brotherly love. So the head is hydrophilic, and then it has that same glycerol backbone that our triglycerides have. And that's where the oxygens are. So we're going to keep those in that head. And then the phosphate group also has all of the phosphorus, nitrogen, and oxygen. So keep all of those electronegative atoms together. And then we are going to have the two tails, and they are hydrophobic, just like they would be on a triglyceride. So there's two fatty acid chains, and they can be both saturated, both unsaturated, or a combination. All right, so they are found in cell membranes. So every animal is going to have this all throughout their cells, but they're also found in some other places. So one of the places we get them for ingredients in food is soy, and another is egg. And so a lot of times when you read labels, it won't just say lecithin, which is the specific example of what this is. They'll say soy lecithin or egg lecithin. So lecithin is a specific version of this phospholipid. And for our purposes, we'll just kind of use those interchangeably. Not all phospholipids are lecithin, but lecithin is always a phospholipid. So it's just kind of a specific case. And it's also found in milk. Now, milk has fat in it. So if you get milk directly from your cow or your goat or your sheep or whatever, then the cream will rise to the top. So you've ever heard that phrase before, the cream always rises to the top. It kind of means that the people who are really good at something, that really excel, make themselves known by standing out from everybody else. And it comes from this idea that when you get milk directly from a cow or sheep, it's going to rise to the top. So remember that the cream has our fats in it, right? And fats and oils tend to be less dense than water. So they're going to float up above the water. So in order to keep them in the milk, the structure of the milk is such that the oils are kind of packaged in these little bubbles, and the bubbles are surrounded by the phospholipids, sort of forming a kind of semi-membrane. And when you see homogenized milk, so remember, homogeneous means the same throughout. What they've done with homogeneous milk is they've made all those bubbles the same size. So if they're all the same size, they have trouble getting together. If there's some larger and some smaller, then the larger ones kind of consume the smaller ones, getting bigger and bigger and bigger. But if they're all the same size or homogeneous and very small, they have a hard time grouping together. And if they have a hard time grouping together, then they have a hard time floating to the surface. Now, all of that's very kind of complicated, but that's just kind of a side note to explain why you see homogenized on things. So most of our milk, whether you get skim milk or whole milk or whatever, you don't have to worry about the cream rising to the top because of this homogenation process. But the point being that the fats that are in milk are contained in these phospholipids. All right, so again, when we draw this picture, we're going to be representing a phospholipid. The circle is the polar part, so it's in love with water. The tails are the non-polar part, so they're afraid of water. They're hydrophobic. All right, so remember I said that when they are near each other, the heads will stay together, the tails will stay together. So if I just have a single layer of them, they might form with all of the tails pointed in, and this is what happens in milk. So all the tails point in, and then we can have some butter fat or milk fat on the inside of this. And then the polar parts are going to point out, and milk is mostly made of water, right? So the polar parts are going to be out here where all the water is. So we call this a meso. We could also have it in reverse if we have something really oily. So mayonnaise is this way, where mayonnaise is mostly oil, so all the fats are on the outside, but there's a little bit of water in there. And so the phospholipids hold the little bubbles of water inside with the phosphorus group surrounding them. Okay. What happens in cell membranes is we get a bilayer. So we have the phosphorus groups pointing in one direction, the tails, and then another set of tails and the phosphorus groups pointing in the other direction. And they all kind of line up in a blob. And that can be very small and be a vesicle. So we talked about the vesicles that hold the neurotransmitters that go throughout the cell. So this could be a small one, or this could surround the entire cell. Now, when it's a cell, then you end up with, in addition to all of these phospho groups, you have the receptors and you have channels and all those other things. Ask your biology teacher about those. I don't know the details about those. But the phospholipids are really important because they help keep one cell separate from the other cell. They keep your cells from leaking out and just being a big pile of goo. You have individual cells, okay? So they're very important biologically, and they're found in foods. And because they're emulsifiers, they're going to help keep foods together. So for example, with milk, that's a very important thing to be able to contain that milk fat in addition to the water. Because for mammal infants, milk is the only thing that they have for their first, depending on what kind of mammal it is, days, months, maybe even years of life. They're primarily drinking milk. And so it has to have the water, the sugar, the fat, the protein, the minerals, the vitamins, everything has to be in there, whether it's polar or non-polar. So having phospholipids is really, really important in a food like milk. Now, for our purposes, since as adults we drink a lot less milk and have more complex diets, we want to have emulsifiers in things that have both fats and waters. So the most obvious example would be things that you have eggs in, like a cake or a bread or cookies. It helps to hold the butter that you put in there or the oil in together with the sugar or the water that you have. This is kind of a close-up of me cells, and just to emphasize in different areas what they might look like. So with milk, most of things are water. So this outside area is water, and the inside is where the oils would be, and we would have a me cell with the phospho heads pointing out. And it would reverse itself to form mayonnaise. So most of the outside is various kinds of oils. And so we'd say that's an organic solvent because water is inorganic. It doesn't have any carbon in it. Your watery stuff is on the inside. So they just invert themselves. And if we have something like a me cell, usually it's not as small as that picture would indicate. Usually you'd have this huge area that was these phospholipids and lots of triglycerides on the inside. So this is more realistic size-wise. You would have a large area surrounding the triglycerides. All right, so one of the things that we do with milk is we put it in coffee, and another thing we do is we make butter from it. And so if you take cream, cream is almost all these oily parts, but it's in the me cells and there's water on the outside and it's mixed pretty well. We want to get all the oils together in order to form butter. And so what we'll do is they talk about churning butter. Basically, you just got to beat it really hard or blend it or shake it or whatever. Sometimes we'll do this in lab. We didn't do it this year. And it doesn't take too long. In just a minute or two, just taking cream, you can beat it until the watery part comes out, as you can see here. This is skim milk. And then this part here are all the oils and fats together. And that's basically what butter is. And so, you know, you would rinse that and maybe add a little salt and things like that. And so we call this skim milk here buttermilk because it comes from making butter. Now, when you buy skim milk at the grocery store, they don't make butter first and then give you the skim milk. And so that's the distinction. Often buttermilk will also be acidified. And so sometimes it's used as an acid to react with food. So we've talked about how acids can be important in food too. Okay, so and then I mentioned coffee. I'm a big fan of having a tiny bit of coffee with my cream and sugar. If you go and have a latte or Americano or all those things that have milk products in them at one of your fancy coffee restaurants, they'll usually froth the milk. And so they'll have some kind of a device that beats the milk, but it's not going to beat it so much that we're going to get the butter pulled out. It's just going to beat it enough to break up those me cells and spread out those fatty parts. And those have a very smooth texture. And so that's going to give the creaminess to the milk. Okay, so we also get some of the fatty parts moving to the surface. and we mix in some air with that. Remember, air is mostly nonpolar too. Now you also get proteins in there. So frothing it includes both fats and proteins. I usually spend a little bit more time on this because it can be kind of complex, but your ability to get a lot of this frothiness is going to depend both on how much fat is there and also on the temperature, because the higher the temperature, the more those oils are going to be liquidy and slide around. But it's also going to change the shapes of the proteins because it's talked about heat denaturing the proteins and things. So there's a lot of things that go into how you build a foam of things. Okay, Chris, you had a question? No, okay, I thought it was awesome. Okay, so I think that's the last slide. So the main thing here is that phospholipids come in a couple of different places, mainly around cell walls, but also in milk and eggs. And that they can serve as emulsifiers to hold food together. So those are kind of big ideas that I want you to get from this. Okay, so we talked last week about vitamins. And again, I usually have time to go into more detail on this, but I'm not going to go into all of this. So the difference between vitamins and minerals, they're both micronutrients, which means we need them in only small amounts. Minerals are inorganic, so they're mostly going to be elements. They're not going to be really carbon-based. Vitamins are organic, so they have these carbon chains and things. And vitamins can be divided into two categories, fat-soluble and water-soluble. And so I want you to know these four are the fat-soluble ones, and you can just know the letters. I have the names here because they're kind of related to some things that we're going to see, but you just need to know which letters are fat-soluble. So because they're fat-soluble, they can get stored in the fatty parts of our body. And if you consume a lot of them on a regular basis, you can have an overdose of them. So when you watch Vitamania, he talks about what happens when you have too much vitamin A. Now, you can also have too little, and he talks in the video about that as well. So these are things that you don't want to just say, oh, I'm going to have as much of this as possible because more vitamins is better. That's generally not the case with fat-soluble vitamins. You want to have the right amount and be good. Now, if you have double the amount of vitamin A you need today, but then you don't have vitamin A for a few days, that's okay. What you want to avoid is having a lot of extra vitamins every day. And that's where doctors are now starting to caution people about maybe you don't need a multivitamin every day. Maybe you need to think about what does your diet look like and whether or not you need to have that. Okay, so we want to look at what some of these things do. Vitamin A is half of a beta carotene molecule, and it's called retinol. What part of the body does the word retinol make you think of? The eyes, yeah. And you may have heard that vitamin A is good for your eyes. It is very good for your eyes. And so that retinol makes me think of retinas. And so that's why I have that there, so you can see that connection. And then vitamin D helps your body to incorporate calcium into your bones. And so this calciferol here helps me to think, oh, okay, it has to do with calcium. The other two, the names don't really fit in with something that you would know, but you will see tocopherol on ingredient labels sometimes. And so we'll take a look at that in a little bit. Okay, I guess that was all I had on that. So yeah, there's the potential for overdose because you can store them. This one is good for eye health. Retinol is found in orange foods. Beta carotene is a very orange color. So carrots, sweet potatoes, some in tomatoes, though they also have a red coloring in them. Cantaloupe, those kinds of things are going to have vitamin A. But it can also be covered up by other colors. So your dark leafy green vegetables like spinach has a lot of vitamin A, but it's also got a lot of chlorophyll. So the chlorophyll kind of covers up the color. Okay, one of the things that's really important when talking about vitamins is the bioavailability. And this is another place that vitamin supplements can not be as helpful as you might think. And it really kind of depends on which supplement that you're looking at. It all varies from one thing to another, one particular vitamin to another. So bioavailability is talking about how readily your body can use it in the form that it's in. So sometimes it can take it from a particular kind of food very easily, but not from another kind of food. So things like, was the food frozen? Was the food cooked? Those kinds of things can really affect bioavailability. So in one of the movies we watched early on acids, they talked about nixtamalization. So nixtamalization was taking the corn and soaking it in a base, in lime. And it turns out that a lot of the B vitamins that we'll get to are in that hull, the hard outer part of the corn. But your body can't use it if you just eat that corn straight. So soaking it in the lime is what makes those nutrients available. When white people came over from Europe and started saying, hey, here's this, food that's available here, let's try growing this, we kind of like it. They didn't really like that whole soaking it in lime process. And for whatever reason, they didn't use that. And so they started developing vitamin deficiencies because they weren't soaking it in the lime, soaking it in some alkaline substance. And so that's why that nixtamalization was really important. It's not really for the flavor of the food. It's really more to make the nutrients available. So when we talk about our fat-soluble versus our water-soluble, our fat-soluble vitamins are going to be more readily bioavailable if they're in oils. This is a picture of some grain, rice, that has been grown to have extra vitamin A in it. They call it golden rice. And they particularly ask people to grow that in areas where there's a lot of vitamin deficiency to help protect the eyesight of people in those areas. Sometimes on food labels, you'll see vitamin A is listed as vitamin A palmitate. Palmitate is palm oil. And so basically that means that they have the vitamin A there in an oily form, so it's more bioavailable. All right, I'm going to skip through some of these. Rickets is a disease that people get if they don't have enough vitamin D. You can see their legs are kind of bowed out here or bowed in, and that's because they haven't been forming the calcium there. So that's kind of the connection there. We can produce some vitamin D with sunlight. So our body can produce some of it. So usually we say vitamins we can't produce ourselves. Typically we'll say that with vitamin D, at least at certain times of year, you need food to get you some of that vitamin D. So people from northern climates are particularly susceptible to this. And the reason for that is that in northern climates, you have much of the year where the sun is only out for a few hours a day and the sun is at a very low angle. And so you're not getting a lot of direct sunlight. And so lower levels of sunlight mean that there's less time for your body to produce this vitamin D. Now, when the sun is directly overhead, it's going through a lot less atmosphere. It gets filtered out a lot less. So if you live near the equator, then even if you're only going out 15 minutes a day, you're still getting enough sunlight in order to produce that vitamin D. And that's not true in the northern climates. Okay, so water-soluble vitamins, this is all the B vitamins and C. So B and C are water-soluble ones, and they're not easily stored because most of your body is water. They're just going to get washed out. So as long as you're drinking enough, you probably can't easily overdose on the water-soluble vitamins. Now, that's not something to test. Don't try and have, like 10,000 times the amount that you're supposed to have. But mostly, you can consume these in large quantities, and they'll be washed through whatever isn't used. But this also means that you can't just, oh, I'm going to stock up by eating a whole bunch of oranges on the first of the month and I'll be good for the rest of the month, because you're not going to have enough vitamin C to last you that month, since it's all going to be washed out. You have to eat it regularly. And so this is why things like long sea voyages, particularly in the 16, 17, 1800s, when crossing the Atlantic was a big deal, why that was problematic, because you can't keep fresh foods on board for months at a time. And so that makes it difficult for people to consume those. And with vitamin C, it tends to degrade over time, particularly with heat. So if you tried to boil off the lemon juice or lime juice and just make small amounts that you could carry with you very easily, you're going to destroy the vitamin C. If you try to dry the fruit, you're going to destroy the vitamin C. If you try to cook the food, cook it into meals and things, you're going to destroy the vitamin C. It has to be fresh. And so that was one of the problems of these long sea voyages. And so, yeah, it's the B and C vitamins. The main thing I want you to know about the water-soluble ones is anything that's a B vitamin and C are going to be water-soluble, and you have to have them on a regular basis, but you're not likely to overdose. We're going to look at some food labels here in a few minutes, and so some of these names are going to show up. These are the main ones that you see, but there are a few other B vitamins, and they're just so common that we don't really worry too much about them. Some of them are associated with specific diseases, beriberi, pellagra, that kind of thing. Folic acid, they'll talk in the movie about, helps when a fetus is being developed early in pregnancy to close the neural tube, the spinal cord. And so they'll talk a little bit about what happens if somebody does not have the appropriate amount of folic acid. Now, because the spinal cord is developing early enough that a woman may not know she's pregnant or just realize she was pregnant, it is really advised that anybody who's trying to get pregnant take a pregnancy vitamin. And that in particular is going to have some extra folic acid in it, just to be sure. And so if you hear about somebody taking neonatal or pregnancy vitamins, that's where that comes from. The other one I want to focus on is cobalamin, and that's because there are no plant sources of cobalamin. It only comes from animals. And so what that means is that if you are a vegan, which a vegan does not eat anything from any animals, they don't eat eggs, they don't eat milk, they are going to have to take a vitamin supplement of the B12. If you don't, then long-term deficiencies of the B12 can lead to dementia. So you don't want that. So it's okay to be vegan, but if you do, please take a multivitamin.